In this video, we will learn about osmosis. Osmosis is the movement of solvent molecules from a solution of higher concentration of solvent molecules to the one with a lower concentration of solvent molecules through semi-permeable membrane. Now, semi-permeable membrane will allow solvent molecules to pass through it but not the solid molecules. In cells, it is movement of water molecules from a dilute solution to a concentrated solution. Depending upon the movement of water into or out of the cells, osmosis is of two types. First, endosmosis. It is the inward movement of water into a cell through a semi-permeable membrane when it is placed in water or solution which is less concentrated, that is dilute solution, than the cell sap. So if the solution is dilute, that means it is having higher concentration of solvent. It causes swelling of the cell. Exosmosis. It is the outward flow of water from a cell through a semi-permeable membrane when it is placed in a solution which is more concentrated, that is, has a lower concentration of water as compared to the cell sap. That means we know that the concentrated solution will have lower concentration of the solvent. It causes shrinkage of cell. Then examples of exosmosis and endosmosis. When some resins are kept in water for some time, they swell up because of endosmosis. In this case, water from outside diffuses through the semi-permeable membrane of resins. Influx of water in resins builds up a pressure which after reaching a limit bursts the resins. On the other hand, when grapes or swollen resins are kept in strong solution of sugar, they shrink due to exosmosis. So here we can see that this is the dilute solution and this is the concentrated solution due to the presence of sugar molecules. Now we know that concentrated solutions will have less concentration of solvent or water, and dilute solution will have more concentration of solvent, that is water. So maximum water molecules will move in this direction, that means from dilute solution to the concentrated solution to this semi-permeable membrane, and the sugar molecules, they are not allowed to pass through it. Now let us perform an activity. Aim of the activity is to demonstrate osmosis using cellophane paper. Cellophane paper is a semi-permeable membrane. Procedure. Fill a thistle funnel with 25% concentrated sugar solution and cover its mouth with a cellophane paper. So this is the cellophane paper covering the mouth of the thistle funnel. Invert the thistle funnel in a beaker containing water and suspend it. So this is the beaker and it is containing water. Mark the level of sugar solution in the stem of the funnel. So the level is marked and the level of water in the beaker. So this is also marked. Leave the setup for about 2 hours. Repeat the same experiment by taking plain water in the thistle funnel instead of sugar or salt solution. What is the observation? The level of sugar solution in the thistle funnel rises and the level of water in the beaker drops. In this case of plain water, no change in the level of water in the thistle funnel or beaker is seen. So that means we can see that from so we can say from beaker there will be inward flow of water to the thistle funnel. So what is the inference? Some water from the beaker has passed through the cellophane paper into thistle funnel containing the sugar solution. Since the cellophane paper will act as the semi-permeable membrane, allowing only water molecules but not the sugar molecules. Sugar from thistle funnel has not passed into the beaker. The cellophane paper acts as a semi-permeable membrane. It allows water molecules to pass through it but not the sugar molecules. Then, how long does osmosis continue? Theoretically, the process of osmosis should continue till the concentration of water molecule is not the same on two sides of the membrane. In the experimental setup in activity, water from the beaker diffuses into the thistle funnel through cellophane paper, which acts as semi-permeable membrane, causing rise in the level of sugar solution. The rising of level in the stem of thistle funnel causes increase in the pressure or weight of column of solution. The increase in weight of column, that is column pressure, reduces further osmosis. A stage comes when no osmosis occurs even when the concentration of water molecules is not the same on the two sides of the membrane. In the state of equilibrium, the water molecules from the beaker tend to force upward through the semi-permeable membrane, but the weight or pressure in the column from above holds them outside the membrane. Now, the significance of osmosis. It helps in absorption of water by the plants, provides turgidity to the cells, develops and maintains turgor pressure, allows the movement of water from one cell to another. Opening and closing of stomata is regulated by osmotic entry and exit of water in gut cells. Resistance of plant to drought and frost is brought about by osmosis. Many plant movements such as folding and drooping of leaves are brought about by osmosis. I hope this topic is clear to you. Thank you.